All right, I'd like to welcome everyone to my thesis presentation, where I investigated algal overgrowth on scleractinian coral in light of environmental as well as biological parameters around Dahab in Egypt. Now, first, a quick introduction to what I like to call the benthic war of coral reefs. As you're probably all aware, coral reefs are incredibly diverse, and usually with great diversity comes fierce competition, which is not different in coral reefs either. There's a bunch of different players, so to speak, involved in this, and I'm just quickly going to introduce the ones of importance to my study, which, of course, on one corner, we have scleractinian or hard corals. They can exhibit a wide variety of growth forms, ranging from quite simple structures like encrusting or massive dome-shaped colonies to, as depicted here, more complex structures like this branching coral. And then on the other corner, of course, we have algae, which are very often effectively summarized by functional groups, namely crustose coralline algae, or short CCA, algal assemblages, which are mostly multi-species assemblages of filamentous turfs, and larger macroalgae. Now, algal abundance in a reef is generally governed by two processes, namely top-down control through herbivores and bottom-up control through nutrients. Now, if there's a reduction in herbivore abundance or an increase in nutrient concentration, or a combination of both, especially over extended time periods, this can lead to a so-called phase shift, in which the corals are being replaced as the dominant life form of the reef by algae, which usually has dire consequences with drastic loss of diversity and ecological functions of that system. Now, coming to the location where I conducted my study, as mentioned, I was in Dahab, situated in the northern Red Sea in the Gulf of Aqaba. It's a relatively small city with around 15,000 people, and it's been a famous diving destination ever since the 1990s. Now, Dahab has been developing rapidly ever since then, especially the tourism sector, which is the main source of income by far there. And unfortunately, a lot of other sectors did not manage to keep pace with that development, including sewage treatment, which is highly inadequate. Important to note here, it's quite sensitive information and quite scarce, difficult to get to, but it's been reported already 15 years ago that this was an issue in Dahab, with around 60% of the buildings not being connected to any kind of facility. And there's not a sign of improvement of the situation, quite the opposite, actually. If we look at satellite imagery from 2013, these black areas that you can see in the middle are sewage treatment ponds. By 2016, I'm not sure one of them might still be active, but definitely by 2018, they've been completely desiccated. And they remained dried out ever since, also during the sampling period when I was there in January, February. Now, if we look at what's been happening underwater in Dahab over those years, actually, and I'm going to show you data from the Reef Check program conducted by the Red Sea Environmental Center. And as you can see, algal substrate cover has been steadily increasing at most of the dive sites, both at 10 and 5 meter depth. Which also brings me to the aim of my study, which was to investigate algal overgrowth on corals, both in terms of the frequency as well as the algal functional groups involved. Today, I'm only going to focus on the frequencies, though, and to explore potential relationships of overgrowth with anthropogenic stressors, purely environmental parameters, as well as biological characteristics of the organisms involved in these interactions. Now, I chose 7 sites to sample, 4 to the north of Dahab, 1 in the center, and 2 dive sites in the south. First, I categorized tourist pressure for each site, which is basically how many people are visiting that site, so snorkelers, divers, as well as swimmers, as well as the water exchange rate of each site, given that this has an impact on the nutrient retention time, of course. I did so by handing out questionnaires to the local dive guides and just asked them to categorize the sites for me, as they had quite the experience. Furthermore, since I was not allowed to take any kind of physical samples, yet alone water samples to measure nutrient concentrations, I drafted a sewage input index equation specific to Dahab, where I utilized Google Earth as well as personal communication and observations to try to get as good as possible categorical estimate for each site. This equation considered the building density of the surrounding area, the capacity, as well as the current occupancy rate of the hotels. Furthermore, the biological variables were collected by means of scuba diving with randomized quadrats along a transect, both at 5 and 10 meter depth. These variables included everything from coral genus to growth form to the colony size to algal functional groups if it was overgrown, the coverage of this overgrowth, the tissue state underneath, and so on. So I basically tried to collect as much data as possible underwater. Now, this data was then analyzed using R and SAS, and I applied log linear models with a binomial response of presence and absence of overgrowth. I ran three main models. The first one looked at spatial differences, so looked if there were differences between the dive sites or differences in depth. The second model considered all the environmental parameters, so the water exchange rate, the tourist pressure, and the sewage input index. And the third model looked at the only biological variable that turned out to be worth investigating, which was coral colony growth form. Important to note here is that I simplified it into two groups, as I mentioned in the introduction, as in complex growth forms like branching corals and more simple growth forms like encrusting or massive dome-shaped colonies. All right, now coming to the results. So I sampled a total of 873 corals, 127 of which were overgrown or at least in contact with algae. Regarding the models, the first model did not yield any significant differences. The second model, however, indicated a mild effect of the sewage input index alone, with more overgrowth at sites with low sewage input. And this third model was highly significant, with complex, or namely branching corals, being much more frequently overgrown than their encrusting or massive heterospecifics. Now, in regards to my discussion, I would like to discuss 3 points here. The first point is actually not directly related to my results, but important nonetheless, which is that algal abundance during the time of sampling was incredibly low, actually. If you recall the graph that I showed quite at the beginning with this ever-increasing algal substrate cover, this is what the graphs actually look like with the data that was collected in January, February incorporated. And as you can see, most of the sites had vast decreases since last year of algal abundance, which obviously also impeded further detection of additional effects for me, given the relatively low sample size. Now, coming to potential explanations, what could have caused this, coming back to bottom-up and top-down control. So first, bottom-up control would basically imply that there was a reduction in tourism or sewage input. And I've already emphasized that a sudden improvement of sewage treatment is highly unlikely. There's not a single indicator suggesting that. Furthermore, tourism industry in Egypt collapsed at around March, beginning of April. So in January and February, there were still as many tourists in Dahab as ever, and therefore also producing as much sewage as always. So therefore, bottom-up control in this regard is quite unlikely to have caused this. On the other side of the spectrum, could this be explained by the herbivore abundance? Now, I will go to show you the data also from the reef check program. You can see the herbivores, fish on the left side and the urchins on the right side. If you focus on the left side, you see that it appears that most of the population appear to be increasing or recovering for the last 2 or 3 years more so. It's important to note here that these are purely sums of herbivores which are monitored by the reef check program, and so some species are missing, and obviously they're not weighted in terms of their actual consumption rate. But therefore, this does not necessarily represent herbivore pressure, but nonetheless, it may be an indicator of the fact that herbivores may have been a contributing major factor to the low algal abundance that has been observed now. So this is definitely possible. The second point, which you may have already been wondering about, is that there was more algal overgrowth at sites with low sewage input. Now, not surprisingly, this is contradicting previous studies. On one hand, Naumann and others, who in 2015 conducted a study in Dahab and found significant correlations between nutrient concentrations and algae coverage. And on the other hand, well, basically the entire scientific consensus on the relationship between eutrophication and algal blooms. Which basically leads me to believe that this result was most likely due to inadequate methodologies, partially indicated by having found only a mild effect with a relatively high p-value. Furthermore, the locations of the actual sewage inputs remain unknown. I could not identify any sewage pipes, they're quite well concealed. And my index assumes that they go directly to the ocean, but they might as well go 2 kilometers north and then feed into the reef. Furthermore, the effects of sites may overlap, and the sewage can just be washed away to another site, further impeding detection of any differences. So in conclusion, what I find most likely here in this case was that the sites have been grouped rather arbitrarily in accordance to a non-representative index, and then spatial fluctuations just kind of generated this mild effect simply by chance. The last point that I would like to discuss is also a result that I'm much more confident about, which is that there was significantly more overgrowth on complex corals, or branching corals mainly. Again, this is actually contradicting previous studies, although there's just a handful of studies on this matter. And the last two authors listed here, Sewage and Vermeij, excuse my pronunciation here, suggested that corals are actually subject to what is called the resource availability hypothesis, which basically states that there's a trade-off between investing into competitiveness or investing into avoiding competition altogether. Now, all these studies have shown that the corals that I call simple, so encrusting and massive colonies, they do have a lot of contact with the benthos, and they grow quite slowly, actually, and they therefore encounter algae quite frequently. They also invest a lot into defense mechanisms, and when they encounter algae, they usually win those interactions. In contrast, branching corals, their strategy is to grow fast and grow up, basically avoiding anything that's crawling on the benthos. Now, technically, these corals have a lower frequency of encounter, but when they actually encounter algae, they usually lose this interaction because they don't have the defense mechanisms. It's important to note here that my results only partially contradict this hypothesis, because I can say, okay, I've found the opposite patterns in terms of frequency, but I cannot make any conclusions on the competitive outcome because I only took single samples and did not take repeated measurements of the same interactions. So I cannot actually say who was winning this interaction and who was losing. Nonetheless, there's two hypotheses that I developed in potentially explaining the patterns that I found. The first one I dubbed the low flow hypothesis, which basically states that the reduction in water flow within the coral structure may facilitate algal settlement and their competitive mechanisms. There's a bunch of supporting evidence for that. Several different algae have been found to prefer low flow regimes. And furthermore, their mechanics to damage corals, like inducing hypoxia and utilizing allelochemicals, are a lot more efficient if there's a low flow environment, obviously. Also, I then ask myself, well, if this is actually the case, shouldn't be this pattern a lot more obvious at sites with a strong current, or how I adopted in my thesis a high water exchange rate? So I ran a chi-square test on the two different proportions, and indeed, actually, the complex corals contributed significantly more to total overgrowth at sites with a high water exchange rate. So strongly supporting my hypothesis here. Of course, there's some loose ends to it. For example, CCA, so crystallose coralline algae, are generally under-investigated in this regard, and I could not find anything in the literature in terms of flow preferences. And most notably, since this hypothesis does not really consider locally restricted conditions, but is actually quite broad, it kind of implies an occurrence on a global scale. And given that I appear to be the first one who actually reports this, this should definitely be subject to further investigation, whether this could also be the case in other locations of the world. The second hypothesis that I would like to bring forward is what I call the herbivore refuge hypothesis, which basically states that with the recovering herbivore populations around Dahab, branching corals provide algal refuge from larger herbivores. Again, there's some supportive evidence for that. It's been shown that herbivory rates are reduced within branching corals, as well as some algae actually having elevated production there. And most notably, the increases of overall herbivore abundance over recent years that we've seen from the reef check data. Which also brings me again to the loose end of this hypothesis, and I've mentioned it before, total herbivore abundance does not equal to total herbivore pressure. So in order for this hypothesis to verify, one first have to verify that the herbivory rates have actually increased over the last 2 or 3 years. Obviously, these two hypotheses are not mutually exclusive. Now, in conclusion, first of all, I did not detect any biologically relevant correlation between sewage input and algal overgrowth on corals. As I mentioned, most likely due to inadequate methodologies or non-representative index. And it just highlights the importance of being able to actually take physical samples that can be measured accurately in order to make any studies on eutrophication effects. Furthermore, detection of ongoing ecological changes such as phase shifts was not possible. Maybe if one of the sites would have been completely pristine, I would have been able to make some comparison, but like that, with no repeated measures of the same interactions, I do not know about the competitive outcome. And third, most prominent effect by far was growth form. However, given that this pattern has not really been described before, the temporal and spatial consistency of my findings would require further verification, of course. It is partially contradicting with the resource availability hypothesis. It would be interesting, though, how far this contradiction actually goes. And yeah, water dynamics as well as herbivory may be potential factors here. And these findings may have potential implications for coral conservation or restoration efforts, as the relationship between current dynamics and the coral growth form may affect the coral's susceptibility to algal overgrowth. So yeah, that was it from my side. Thank you for tuning in, and I'll be happy to answer any kind of questions you might have.